Hello and welcome. So, cerebellar cortex layers and Purkinje cells and the anatomy surrounding it so that you can answer MCQs. I do not think that you should rote memorize this or use mnemonics to memorize this. It is better to just understand it and it will just commit to your memory. And this is not exhaustive, okay? This is just a small topic. Um, it's not a small topic because it is not exhaustive video. It's just going to help you understand and answer MCQs. So the whole discussion in the cerebellar cortex is around Purkinje cells. This is the diagram, and then I made a spilled spaghetti version of it. So yeah, you have three layers in the cerebellar cortex. You have the molecular layer, which has the basket and stellate cells. You have the Purkinje cell layer, which has the Purkinje cells, and then you have granular layer, which has the granule cells and the Golgi cells. Now, the only output, the only thing that goes out of the cerebellar cortex is Purkinje cells. It is always inhibitory, and it is to the deep cerebellar nuclei, which are dentate emboliform globose vestigial from lateral to medial, deep cerebellar nuclei. Okay, doctors eat good food, dentate emboliform globose vestigial. Doctors eat good food is the mnemonic. Now, Purkinje cells are the only output, okay? Only output of the cerebellar cortex. You know that cerebellum is responsible for fine tuning and course correcting while the movement is in progress. Okay, so we need to understand all this so that we can know how your body, how cerebellum course corrects or fine tunes while the movement is in progress. So you have Purkinje cells, which is around which the entire discussion is based. So you have contralateral inferior olivary nucleus that sends its climbing fibers directly to the Purkinje cells. So you have to remember one more thing, which is anything that is going up is going to be excitatory and is going to use glutamate. Anything that is going down within the cerebellar cortex is going to be inhibitory and going to use GABA. So look at climbing fibers, which are from the contralateral inferior olivary nucleus. They go directly to your Purkinje cells and they are stimulatory. They use glutamate, they are going up, climbing fibers. Okay, so that is stimulatory. Then look at mossy fibers, which comes from pontine nucleus. They are not going to go directly to the Purkinje fibers, but to the granule cells of the granular layer. And they are also stimulatory because they are going up. These are all inputs to the cerebellar cortex. Okay, the only output, what is the only output of the cerebellar cortex? Purkinje cells, which is always inhibitory. It uses GABA. Who does it give it to? Deep cerebellar nuclei, dentate emboliform globose vestigial, lateral to medial. Okay, now mossy fibers from the pontine nucleus are going to go up and stimulate the granule cells. Granule cells are going to go up and stimulate the basket cells and the stellate cells of the molecular layer. Now there is also inhibitory interneurons. Look at Golgi cells. Golgi cells are inhibiting the granule cells. This is happening within the granular layer. Okay, so Golgi cells are going to inhibit the granule cells. But granule cells are going to send its stimulatory input to the basket and stellate cells, and then basket and stellate cells are going to send their inhibitory output to the Purkinje cells. They're going down, that's why they are inhibitory. They're going to use GABA. So basket and stellate cells are going to inhibit Purkinje cells. And after all this, Purkinje cells is the only output, always inhibitory, going down, so always inhibitory, to your deep cerebellar nuclei, which are arranged from lateral to medial as dentate emboliform globose vestigial. Now, because we are outside of the cerebellar cortex in the deep cerebellar nuclei, now your dentate emboliform globose and vestigial are going to project fibers through the superior cerebellar peduncle, which is excitatory, but this is outside of the cerebellar cortex. Within the cerebellar cortex, anything that is going up is stimulatory, anything that is going down is inhibitory. And vestigial is going to use your inferior cerebellar peduncle. Okay, now if they ask you to recognize the Purkinje cells, so look at the Purkinje cell layer, okay? The Purkinje cell layer, and above it is this molecular layer. Molecular layer is going to have your basket cells and stellate cells. Then you have the Purkinje cell layer, okay, Purkinje cell layer, and then below it is the granular cell layer. That is the granular cell layer, which has the Golgi and the granule cells. Now here it looks more distinct. It's flask shaped. You can see, right? Flask shaped Purkinje cells. Look here, flask shaped Purkinje cells. Okay, yeah, so that's how it's going to look. And then if they give you this image, you can see flask shaped Purkinje cells. Okay, this, that is your Purkinje cells, and that is the Purkinje cell layer. Above it is molecular layer, and below it is granule cell layer. Thank you for watching.